Welcome to the Biological Life Sciences students. Today in this video, we are going to see how to download a gene or a protein sequence from NCBI's database. That is nucleic acid database or a protein sequence database. Okay. So first of all, we will go to the address bar and we'll type a URL that is www.ncbi.nlm.nih.gov.in. Okay. So when you press this URL, you come to the web interface of NCBI. This is the official website of United States of government. So let's see first of all how we are going to download a nucleotide sequence. Okay. So here you see a window called as all database. Okay. So in that window for downloading a nucleotide sequence, you have to first select nucleotide. Okay. So here nucleotide is there. We have selected nucleotide and we are going to put the name of the gene. You can put the name of the gene, name of the author. You can put the accession number of the gene that is available or the DNA sequence which you want to download that is available. Okay. You can put the keywords. Okay. You can do various kinds of search in this search bar. Okay. So I will be showing you how to download a gene sequence and how to download a protein sequence from this NCBI's database. Okay. So let's put an example. Say example insulin I am putting. Okay, I'm searching insulin. So when I search this, I get lots of sequences as a result. Okay, so you can see there are lots of sequences that I am getting as a result. uh, so here Homo sapiens insulin is at the sixth number. Okay, you can search any gene if you want. Here Homo sapiens gene is there. So I will be opening this. Okay, so see here when you open this, it opens up into certain kind of format. Okay, so this is a format that is actually called as gene pep format. Okay, so whatever the format you are seeing on the screen is actually a gene pep format, where it starts with the locus, then definition, then accession, that is accession number. Okay, you can see the accession number given over here. Okay, then version of the sequence is there, keywords, source, organism, yes, and the total classification of the organism is also there, then references, author. So this is a gene pep format or a format of gene bank entry. You can see whole the entry of the sequence is there, and at last the origin and the sequence is given here. Okay, so this is the sequence that is given here. Okay, now this is the gene pep format as I told you. Okay, now let me open it in a FASTA format. Okay, so when I click on the FASTA icon here, the sequence opens up in a FASTA format. Okay, so you can see the sequence opening up in the FASTA format. Okay, so why we open FASTA format? Because you get only the sequence. Okay, for example, if you want the sequence for phylogenetic analysis or certain kind of secondary structure prediction. Okay, so if you want to do such kind of analysis on the sequence of the gene that you have downloaded, in this case I have downloaded the insulin gene, you can copy the sequence here. Okay, so see, I am copying the sequence here from the starting arrow till the last nucleotide. Okay, you can see it is ATGC. ATGC means it's a DNA sequence. Okay, so I have copied the sequence from the starting till the end. Now I'll minimize and I'll create a new notepad image or a text document. Okay, you can create a word pad or a word image also. Okay, and I'll paste the sequence here. Okay, you see I have pasted the sequence here. Now this sequence can be used for various kinds of analysis. Okay, so this is the sequence for insulin that I have downloaded. Okay, so you can calculate the number of A here, number of G. Okay, and so on. You can confirm the Chargaff rule also. Okay, so lots of analysis can be done using this sequence. Okay, so similarly this is how we have downloaded the protein sequence. Now we are going to download nucleotide sequence. Sorry, now we are going to download the protein sequence. Okay, so for protein sequence downloading, you have to click on protein here and you have to put this keyword or accession number or the name of protein, etc. Okay, similarly we are going to put insulin only, so that we are able to find out the difference between the two. Okay, so I have put the search put the word in the search as insulin. Okay, and I have searched it. Okay, so what will happen, it will show me the sequence of insulin in protein form, not in gene form. Since I have selected the protein here, so it should show me the sequence in the insulin form. Okay, so let's see how it does. Okay, so see here, there are lots of results that have been shown to me. First of all, Octodon Degas insulin gene is there. Then lots of insulins are there. We are going to see the insulin from human being or any kind of insulin also you can see. Okay, see here on the 14th number, insulin from Homo sapien comes. It is a 110 amino acid protein. Okay, let me open this. Okay, so when I click on the insulin for Homo sapien, I get accession number also. I get lots of information that is opening up here. It will be in single letter code of amino acid. Okay, so for that purpose you should know or you should remember the single letter code of the sequence that is there. Okay, so this is how it opens. See, it's a the accession number of for the protein sequence is there given. Then GI number is also given. Okay, so gene bank ID is given. Okay, so similarly, it opens up into a format like this. Okay, where the sequence is given at the last. Okay, you can see the sequence here is given in the amino acid single letter code. Okay, so you can download this sequence from here. You can open this in a FASTA format so that you can copy the sequence. Okay, so I have opened the sequence in the FASTA format now. You can see the sequence is going to open in the FASTA format now. See, this is the sequence. I can copy the sequence from the top till the bottom, and this is the sequence for the amino acid, and I'll put the sequence in a WordPad. Okay, as earlier I have pasted the sequence in the text document here. I have pasted this protein sequence also. Okay, so you can see how small the protein sequence looks like, just because it is a proteinic sequence, that is a single letter code of amino acid, and this is the sequence for the gene of human insulin, and this is the sequence for the protein of human insulin sequence. You can find using this single letter code by pressing Control F. You can find the number of amino acids, that is polar amino acids. You can find out sulfur containing amino acids. Okay, you can find out parent amino acid. You can find out the simplest amino acid, that is glycine. Okay, then aromatic amino acids, number of aromatic amino acids present here. Okay, so if you want to determine the presence of sulfur containing amino acids in this, so what you can do, you can press Control F and then you can put the code for methionine. Okay, so when you put M, it is going to search for methionine. Okay, so here see first arrow, first amino acid itself is methionine. Okay, you can see it is showing that the first amino acid itself is methionine. Then again when you press enter, again the next methionine. Okay, so likewise there are only two methionines present in the gene. Okay, so you can likewise search different kinds of amino acid likewise, and this is how you are to download a sequence, that is a protein sequence or a gene sequence from NCBI's website. Okay, you can use the sequence for various analysis that will be taught to you on this channel in the subsequent videos, that is coming videos. Okay, thank you for watching, students. I hope you have understood how to download the sequence. Thank you so much.